Now we're going to talk about the lysogenic cycle. Essentially, the lysogenic cycle has very similar steps as the lytic cycle, but the only difference is the end goal of the lysogenic cycle. The end goal of the lysogenic cycle is to incorporate its genetic information with the host cell genetic information. And you can see why this is smart, because as the host cell replicates itself, so is the viral genome replicating itself, and as a consequence, it's silent at the time when it's replicating, but it allows a rapid rapid replication of the virus genome without the host cell even realizing it's happening. And eventually, once you might be asking yourself, okay, so it's replicating itself, but really it's not having any impact on, it's not expressing any of its genes. And when something triggers the host cell or the viral genetic information of the host cell to trigger its genetic information, then we have the virus entering the lytic cycle. And this is when we will see that the lysosome or the lysogenic cycle is now expressing its genetic information. You will see the entire host cell go through the same process as the lytic cycle. But what makes the lysogenic cycle, why are cells doing this in the first place, you might be asking. And the reason behind that is because if you think about it, the lytic cycle, upon infection, is destroying the entire host cell. And eventually the organism will have no host cells for the viruses to infect. And because of that, through an evolutionary perspective, it's very helpful that these viruses go through the lysogenic cycle instead. Because look, the host cell is still alive, and because it's alive, it can still make sure that it's making sure that its host cell is living, and it's actually doing relatively fine, so it can keep on replicating itself compared to having no host cells to infect. So now let's start off with this quick question that we left off in the last video. This question over here is asking us 35S labeled cysteine, 32P labeled phosphate is in new generation. So what this question is asking over here is, we have the first generation, which is radiolabelized, but then what do you expect the next generation, what aspects, or what will this cysteine, the protein, or the element, the phosphate of the DNA, the genetic information, be labelized in the next generation? And the answer to this question is given off by our understanding that, hey, look, protein is not responsible for the passage of genetic information. It's actually the DNA. So right over here, we see that the phosphate 32 will be passed on to the next offspring. And the answer does not reflect that, but please understand that I was talking about the phosphate, not the cysteine, because it's an amino acid. The amino acid is just left on the outside. So think about it, the protein aspect is left on the outside of the host cell. It just injects the viral genome and it just floats off. It doesn't have to do anything else with the virus. Then we have the genetic information inside the cell that will cause the formation of more of the genetic information, first of all, and then also the protein aspect. So we don't have a problem at all. We hopefully understand that now. So the lysogenic cycle, let's get into it real fast. The lysogenic cycle, like we said, the major difference is the end goal. The end goal is to not destroy the host cell. Well, like the lytic cycle, it's just to incorporate itself with the host cell's genetic information. And we also talked about the evolutionary standpoint, and it's just to keep the host cell alive. So here's the overview of the lysogenic cycle. Upon insertion of the viral genome incorporates with host genome. So viral plus host genome is referred to as the prophage. So when we have, like over here, well, first of all, obviously it's going to be, if you would expect this to be entire blue when it's the host cell, that would be the host cell genetic information, obviously. But when we have the viral genome coming inside and incorporating itself with the host cells, then it's called the prophage. So the prophage is kept dormant, it's kept silent. It's not expressing any of its genes. And because it's not expressing any of its genes, there's no obvious impacts, and there's no, you won't even know the virus is there in the first place. It's dormant. and that's the whole reason behind this is, and as like we talked before, the host cell can replicate itself normally without even noticing the virus is there. So phage-encoded repressor binds to the promoter. So we talked about operons in previous videos, the like operon, the trip operon, and just like that, we have operons here. An operon just means an operator. So the phage-encoded repressor will bind to the operator, and this will physically disable the recept or the polymerase required to transcribe the information. This is how it's kept dormant. Every time cell replication occurs, viral genome replicates with it. Later, prophage activates. It will remove itself from the host genome in a process called excision. Excision is, like I said just right now, in other words, is when we have the viral genome now saying it's time to enter the lytic cycle, let's just get out of the host cell. So now it's going to cut itself out of the host cell through a process called excision. And now it enters the lytic cycle. During excision, it is possible host genome is incorporated with a viral genome. What impact will this have on the genome? Now the question is asking us, there's a chance that the host genome will be also incorporated with the viral genome when it's cut off. So let's say that we have the virus and it has a gene for, let's say, lactose or non-lactose metabolism. Let's say it has a gene for let's not give you an example, but the point is, if it has a gene on it that's responsible for doing anything, is giving an extra ability to the virus. So if the host cell gene, which is giving some kind of advantage to the virus, is also incorporated in the viral genome, in the sense that it's taken with it during the lytic cycle when it's activated, it's called a stolen gene. Now genes will move around, obviously. Now stolen genes on the new viral will, once infected on a host, integrate with the host gene of the host. So you're kind of moving around these genes. So hopefully that makes sense. And what I'm trying to get at over here is, obviously we know that the viral genome has to spread itself. After a virus is made, new progeny is made, it will go infect another cell. But if the previous host cell's genetic information, one kind of gene just is incorporated inside, that gene will be delivered to the next genome as well once it incorporates with it. Hopefully that makes sense. And if you remember, we talked about transposons in the other playlist regarding in the molecular biology playlist. You will remember that transposons, we talked about transposons, and we refer to them as scientists think that it's old degraded viruses, degraded particles in our bodies. And because and this is the entire reason why we consider it as viruses, ancient viruses in our bodies, because they're moving around consistently with new genes. So this gives the host cell new ability, an example trait to metabolize lactose. I was about to give that, but this is an example in terms of well, that makes sense. It's not giving the virus itself the ability, but it's giving the new host cell the ability to metabolize lactose. This is why transposons are referred to as ancient viruses in our bodies. In bacteria, this process is called transduction. And we're going to talk about transduction soon. Will a certain gene pass through transduction be expressed during lysogeny? So remember, whatever kind of genetic information enters the host cell during the lysogenic cycle, and the lysogenic cycle, it's incorporating the viral genome inside the host cell. So whatever the genome is, regardless of it being a positive impact on the host cell, it will have a repressor binded to it. So this makes sure that the operator will have a repressor binded to it. This will make the entire genetic information of the virus dormant, regardless of it being beneficial to the host cell. So no, because viral genome in the prophage is repressed through repressors until the prophage genome is attached. Now let's get into the step-by-step process of the lysogenic cycle. So, attachment of bacteriophage and insertion of the viral genome. So obviously we have the virus that's going to insert its viral genetic information inside. The second step is the phage DNA integrates with the host cell DNA. Now we see that the prophage is formed over here. And the third step will be the dormancy phase, when it's just replicating itself without even knowing anything is happening. There are no viral expressions in the organism that's infected at the moment. He's living a happy life. He doesn't even know what's happening. He's gullible and not stupid. He doesn't know what's going on. Then we have the fourth step, which is the activation step. This happens when excision happens, and obviously excision is a process when we have the viral genome, dissociating itself from the host genome and doing its thing, entering the lytic cycle. And then eventually we have so many progeny released inside the environment. Not good stuff, man. Not good stuff.